To dominate the pitch-black, crushing depths of the ocean, the ultimate apex predator doesn't use gills. It breathes air. And while its name might sound like a crude joke, a misunderstanding by 19th century whalers, the sperm whale is actually a highly evolved deep-sea titan. Before it even begins to hunt, it sleeps completely vertically, suspended in the dark like a silent alien obelisk. The name came first. When commercial whalers of the late 18th and early 19th centuries cracked open the massive heads of harvested sperm whales, they found the cavities filled with a white, waxy liquid. They'd never seen anything like it. The substance was named spermaceti, from the medieval Latin spermaceti, meaning whale sperm, because that's what they assumed it was. The species name followed. The substance is actually a highly specialized lipid with extraordinary acoustic properties. The whalers were wrong about what it was, but what it does would take another 2 centuries to fully understand. Set the name aside. The animal underneath, it's Phyceta macrocephalus, the largest toothed predator on Earth, carrying the largest brain of any creature that has ever lived. Roughly 7 to 8 kilograms of neural tissue, about 6 times the mass of a human brain. Males grow to 18 meters and 45 tons. Their lower jaw, narrow and blade-like, is lined with the largest teeth of any predator alive. The upper jaw has none, just sockets to receive them. The sperm whale is a highly evolved, culturally complex, deep-sea titan whose extreme adaptations, including passively collapsible lungs, massive oxygen-storing muscles, and a sonar system producing up to 230 decibels of sound, make it the most formidable predator on the planet. But before all of that, it sleeps. In 2008, a team led by biologist Patrick Miller at the University of St Andrews deployed digital acoustic tags, small instruments glued directly to the backs of sperm whales, and documented something nobody had formally confirmed before. Groups of 5 or 6 whales were drifting just below the ocean's surface, aligned nearly vertically, heads pointing upward, bodies motionless. The whales weren't dead, they were asleep, or something close to it. One group, in an episode now cited in peer-reviewed literature, failed to respond to a vessel passing very close, until the ship accidentally made contact with them. Only then did they scatter. They had been so deeply unresponsive that physical touch was required to break through. Sperm whales spend roughly 7% of their lives in this state. Each episode lasts around 10 to 15 minutes. The exact neural mechanism behind it remains unclear. Popular accounts often describe a half-brain asleep, one eye open model borrowed from other cetaceans. But that specific pattern hasn't been directly measured in free-ranging sperm whales via brain monitoring. What is confirmed is the posture, vertical, motionless, clustered near the surface. A pod of them floating in the dark looks like nothing so much as a cluster of enormous columns, waiting for something the ocean hasn't thought of yet. Then they dive. A sperm whale foraging dive typically covers between 1,000 and 2,000 meters of water column. At that depth, there is no light. There hasn't been any for the last 900 meters. The pressure at 1,000 meters is approximately 100 atmospheres, 100 times what you feel standing in open air. For a mammal that breathes, that pressure creates a specific physiological crisis: nitrogen. When air sits in the lungs under pressure, nitrogen dissolves into the bloodstream at an accelerating rate. Ascend too fast without offloading that gas, and it bubbles out of solution in the veins and joints. Decompression sickness, the condition that can permanently disable or kill human divers. For a creature making multiple dives per day to depths that would be instantly fatal to an unaided person, nitrogen management isn't a side concern.
It's the central engineering problem. The sperm whale solves it with its rib cage. The thorax is structured with flexible ribs and elastic lungs that allow the chest cavity to compress under increasing hydrostatic pressure as the whale descends. The lungs don't get crushed by muscle action. The pressure of the ocean simply pushes them inward, passively, in the same way a balloon shrinks when you squeeze it. At around 200 meters, both the whale's lungs and those of any human diver will have fully collapsed. The trachea and bronchi, the stiff conducting airways, remain open, but the alveoli, the tiny sacs where gas exchange occurs, are shut down. Once the alveoli collapse, nitrogen stops crossing into the blood. Gas exchange ceases. The decompression clock stops. As a 2012 study in the Journal of Experimental Biology confirmed, this alveolar collapse is considered the primary mechanism that limits nitrogen absorption and reduces the risk of gas emboli during ascent in deep-diving marine mammals. The heart slows dramatically. From roughly 60 beats per minute at the surface, the sperm whale's heart rate drops to around 10 beats per minute at depth. Blood is redirected away from the extremities through peripheral vasoconstriction, flowing preferentially to the brain and heart, the organs that can't tolerate even brief oxygen deprivation. Complex networks of arteries and veins called rechia mirabilia, literally wonderful nets, act as biological buffers, helping manage blood pressure and gas distribution across the dive. The whale's confirmed dive record sits at approximately 2,250 meters. One male off South Africa in 1969 held his breath for 117 minutes. Those numbers are formidable. But to be precise, the absolute mammalian depth record belongs to the Cuvier's beaked whale, which in 2014 was tracked to 2,992 meters. Sperm whales are among the deepest diving large active predators on Earth. That's not a demotion, it's just the correct framing. None of this explains how they stay down that long. The lungs are collapsed, nitrogen is locked out, but the animal is still moving, still hunting, burning oxygen at every meter. Where is the oxygen coming from? The answer is in the meat. Mammalian muscle gets its red color from a protein called myoglobin, an oxygen-binding molecule that stores oxygen directly in muscle tissue, releasing it when the surrounding cells need it most. In land mammals, including humans, myoglobin concentrations are relatively modest. Human skeletal muscle contains roughly 4 to 5 milligrams of myoglobin per gram of wet tissue. In marine mammals built for long dives, that number climbs dramatically. Research published in Science in 2013 by Mercida and colleagues analyzed myoglobin concentrations across 130 mammalian species and found that elite divers carry myoglobin concentrations exceeding 100 milligrams per gram of muscle tissue, more than 20 times the human level. Those figures for sperm whales specifically are modeled estimates derived from protein sequence analysis rather than direct tissue assays, but the broader pattern holds across every deep diving lineage studied. Rice University's research communications summarize it cleanly. Diving marine mammals can pack 10 to 20 times more myoglobin into their muscle cells than humans can. The visual consequence of this is striking. Myoglobin is a red-brown heme protein. At moderate concentrations, it gives steak its familiar color. At the concentrations found in elite diving mammals, the tissue darkens past deep red into something close to black. The muscle of a sperm whale, cut fresh, looks almost nothing like the pale or medium red flesh of terrestrial mammals. It's dense, dark, and oxygen-saturated, a biological fuel cell packed into every kilogram of locomotor tissue. There's a further mechanism that makes this possible.
At very high concentrations, myoglobin molecules would normally stick together, aggregating in ways that reduce their function. Diving mammals evolved a solution. Their myoglobin carries a positively charged molecular surface that repels neighboring molecules, preventing aggregation and allowing far higher packing densities than terrestrial muscle could sustain. This electrostatic, non-stick property evolved independently across multiple diving lineages, convergent evolution solving the same oxygen storage problem the same way, again and again.
The blood carries the same logic scaled up. Where human blood runs at roughly 30% hemoglobin by volume, sperm whale blood runs closer to 60%, allowing roughly twice the oxygen storage per unit of blood volume. With the lungs collapsed and inactive, the oxygen in the blood and muscles is the only working supply. In deep-diving mammals generally, around 80 to 90% of total body oxygen stores are concentrated in blood and muscle tissue. The respiratory system, so dominant in land mammals, contributes almost nothing during the hunt. The whale arrives at depth carrying its oxygen supply inside its own body, in total darkness, without a functioning lung. Now it needs to see. The head of a sperm whale is one of the most architecturally extreme structures in natural history. It accounts for roughly 1/3 of the animal's total body length. In large adult males, it can weigh more than 10 tons, and almost all of it, the entire vast interior, is devoted to a single acoustic system. Norris and Harvey, writing in 1972 in what would become one of the foundational papers in marine bioacoustics, described the spermaceti organ, the dominant structure inside that head, as occupying as much as 40% of the entire length of the whale. It's a huge, somewhat flattened, cone-shaped structure. The spermaceti organ holds up to 1,900 liters of the waxy oil. Its outer casing is described in the same paper as covered dorsally and laterally by a thick wall of intermeshing ligamentous cables, many as thick as a man's thumb. But the spermaceti organ doesn't make the sound. This is where the popular account consistently gets it wrong. The sound is made by a structure at the very front of the head, a single pair of specialized soft tissue valves called the phonic lips, or in older anatomical French, the museau du singe, the monkey's muzzle. These lips sit at the anterior end of the right nasal passage, just below and in front of the blowhole. When pressurized air is forced through them, they vibrate and generate an initial acoustic pulse. The spermaceti organ is what happens to that pulse next. The pulse travels backward through the spermaceti organ, reverberating between the large air sacs that bound it on either side. It reflects off the frontal sac, the curved structure adhering to the face of the skull, and bounces forward again. Now it passes through the junk, a dense anterior structure of connective tissue and fat that acts as an acoustic lens. The junk focuses and shapes the energy before directing it outward into the water as a tight, forward-pointing beam. The entire arrangement, phonic lips as source, spermaceti organ as resonating chamber, junk as acoustic lens, functions, in the words of the Hugenberger et al. 2014 anatomy paper, as a unique device to increase directionality and source levels of echolocation clicks in aquatic environments. The clicks that emerge are among the most powerful biological sounds ever measured. In 2000, a team led by Bertil Merle deployed a 5-hydrophone array off Andenes, Norway, and recorded source levels up to 223 decibels, re 1 micropascal at 1 meter. 3 years later, Merle's group published a follow-up study in the Journal of the Acoustical Society of America, establishing on-axis source levels up to 236 decibels, re 1 micropascal at 1 meter. The loudest sounds recorded from any biological source. The National Academy's synthesis report cites 232 decibels as a characteristic figure. In the literature, the commonly cited round number is 230 decibels. That figure needs context. These measurements are expressed underwater, referenced to 1 micropascal at 1 meter. Airborne decibel measurements use a different reference pressure, 20 micropascals, which is 20 times higher, and acoustic energy also transmits differently through water than through air. When you apply the standard conversion, roughly 61 to 62 decibels to account for both the reference pressure difference and the impedance difference between water and air, a 230 decibel click underwater corresponds to approximately 170 decibels in air at the same distance.
A jet engine at close range registers around 140 decibels in air. The sperm whale's click, converted to air-equivalent units, is still louder. But the numbers are measuring different things and can't be compared directly. Any source that places the 230 decibel figure in a direct comparison to airborne sounds without that conversion is skipping a critical step. The click itself is extraordinarily directional. Merle's 2003 study measured a composite directionality index of approximately 27 decibels, meaning the beam is focused tightly enough forward that the same click measured off-axis in a different direction reads 35 decibels quieter. Zimmer and colleagues modeled the forward beam geometry in 2005 and arrived at a directivity index of 26.7 decibels, closely matching the empirical measurement. This isn't a sound radiating outward in all directions. It's a controlled acoustic projection, a searchlight, not a flood lamp. Each click lasts approximately 100 microseconds. Centroid frequency runs around 15 kHz, a range well suited for long-range echolocation on large prey. At that frequency, in that medium, the clicks can detect targets at ranges of hundreds of meters, with the return echo carrying information about the size, density, and distance of whatever the sound struck. Madsen and colleagues, working with free-ranging sperm whales in Norwegian waters in 2002, concluded from the acoustic properties and high output during foraging dives that echolocation is an important sensory cue in prey location. The prey is primarily deep-sea squid, including large, fast species that live in the same lightless zones where the whale hunts. Also on the menu, fish, sharks, and other bathypelagic organisms whose squid beaks have turned up in whale stomachs from Brazil to the Weddell Sea. One hypothesis has circulated for decades that the clicks do something beyond detecting prey, that at point-blank range, an intensity of 230 decibels could physically stun or paralyze a target, making capture easier. It's an appealing idea, and some early papers entertained it. But a 2016 study in Scientific Reports by Feiss and colleagues deployed sound recording tags on free-ranging sperm whales during actual hunts and found something that flatly contradicts the stunning hypothesis. During the terminal phase of a hunt, the buzz, when the whale is closest to the prey, click source levels drop by 20 to 40 decibels compared to the whale's maximum output. The authors concluded that prey capture can't be explained by acoustic debilitation because sperm whales don't expose prey to sufficiently intense sound during the actual capture phase. The sound is for finding, not disabling. At depth, alone in the dark, the sperm whale sweeps its beam through black water and listens for returns. It has been doing this, or something very close to it, for tens of millions of years, since early odontocetes first evolved the pneumatic phonation system that all toothed whales now share. In sperm whales, that system was simply scaled to its most extreme expression: the largest nose, the largest oil-filled cavity, the loudest beam, all packed into an animal big enough to make the whole apparatus viable. The hunt is solitary. The whale descends alone, sweeps alone, catches alone. But the world it returns to at the surface is anything but. Sperm whale society is built around the female. The foundation of their social structure is what biologist Hal Whitehead, in his 2024 paper on sperm whale clans and human societies, describes as the matrilineally based social unit of 10 or so females and their offspring. The members of these units, he writes, form stable, long-term relationships. Males disperse from these units between the ages of 6 and 10, living largely solitary or in loose bachelor associations for most of their adult lives. The females and their young stay together, sometimes for decades. The bond isn't just proximity. Guerreau and colleagues documented in 2013 that calves occupy a specific structural role within these units. They are the central nodes of the social network, the individuals most connected to all others. The primary function of the adult female group, the researchers concluded, is to provide caretakers for calves at the surface while mothers make deep foraging dives. Non-mothers in the unit escort calves, watch over them, and, in some populations, nurse them. A calf's birth is a social event. Its care is a collective project. Female units also share a vocal identity. Sperm whales produce patterned social clicks called codas, rhythmic sequences used in social contexts, distinct from the echolocation clicks used in hunting. Research by Rendell and Whitehead established that units and groups can be reliably sorted into what they called acoustic clans, populations sharing the same vocal dialects across ranges spanning thousands of kilometers. Different clans occupy the same waters but don't crossbreed their vocal traditions. The patterns are passed down maternally, carried in the sound repertoire of females who teach them to their offspring. Cantor and Whitehead's 2015 work showed that different clans also differ in their foraging patterns, movement behaviors, and social interactions, suggesting that the vocal differences are the audible surface of much deeper cultural divergence. Culture in whales is a contested term. Rendell and Whitehead argued for it formally in a 2001 paper in Behavioral and Brain Sciences, defining it as group-specific, socially learned behaviors not reducible to genetics or local environment. The sperm whale fits that definition more cleanly than almost any other non-human animal. These aren't simply animals that behave similarly because they face similar conditions. These are populations with shared traditions, maintained over generations through social learning, that vary from group to group, the way human cultures vary from one society to the next. That social complexity becomes visible under pressure. Pittman and colleagues, in their 2001 paper in Marine Mammal Science, described it during direct observations of killer whale attacks off Mexico and California. The sperm whales appeared largely helpless. Their main defensive behavior was the formation of a rosette, marguerite, heads together, tails out. Adults arranged themselves in a circle at the surface, heads facing inward, flukes pointing outward toward the attacker. The calves are sheltered in the center of the ring. When an individual was separated from the formation during such an encounter, other adults left the circle, moved alongside the isolated whale, and guided it back into the group. The behavior Pittman's team documented wasn't panic, it was coordination, a practiced response that suggests the formation is learned, culturally transmitted alongside the vocal dialects that distinguish one clan from another. The Essex came from the other direction. On November 20, 1820, the Nantucket whale ship Essex, under Captain George Pollard Jr., was working the southern Pacific when a large sperm whale rammed and sank it. All 20 crewmen survived the initial attack. 8 were ultimately rescued. Historians and biologists have since read the incident not as evidence of malice. The sperm whale has no evolutionary interest in killing sailors, but as a response by an animal that had witnessed its pod members being killed by the men on that ship. The whale attacked the vessel, not the humans individually. It targeted the source of the threat with the tools it had: 45 tons of moving mass, a reinforced head, and the biological architecture to survive a collision that destroyed wooden planking. Herman Melville read the accounts of the Essex survivors and wrote Moby Dick. The novel made the sperm whale into a symbol of incomprehensible natural fury. The biology tells a different story: an intelligent animal protecting its family, using the only option available. The sperm whale, it turns out, doesn't just shape its ecosystem through predation. It shapes it through waste. Sperm whales hunt at depth. They consume squid and fish well below the photic zone, the sunlit surface layer where photosynthesis occurs. When they return to the surface to breathe, rest, and socialize, they defecate. And because they've been feeding hundreds of meters down, the nutrients in that waste, iron in particular, arrive at the surface as allochthonous inputs, material that originated outside the sunlit zone and wouldn't otherwise be there. Iron is the critical variable. The Southern Ocean has abundant nitrogen and phosphorus for phytoplankton growth, but iron is scarce. Phytoplankton without iron don't bloom. But introduce iron into that system, and the blooms can be enormous, pulling carbon dioxide from the atmosphere and locking it in biomass that eventually sinks to the seafloor. A 2010 paper in Proceedings of the Royal Society B by Lavery and colleagues estimated that the roughly 12,000 sperm whales then inhabiting the Southern Ocean defecate approximately 50 tons of iron into the photic zone each year. The lead researcher described the fecal plumes vividly as a big plume of diarrhea. The resulting phytoplankton response, calculated using molar ratios from natural iron fertilization events, drives an estimated 400,000 tons of carbon export to the deep ocean annually. Before industrial whaling, sperm whale populations were an order of magnitude larger. The strandings documented in the North Sea since the late 16th century, 168 recorded events between 1560 and 1995, tell a related story. The long gap in strandings between the late 18th century and the early 20th century corresponds exactly to the period of peak commercial whaling in the North Atlantic. As sperm whale numbers crashed, fewer animals wandered those waters. As whaling declined after the International Whaling Commission's moratorium in the late 1980s, sperm whales began reappearing, particularly in the 1970s and again in the 1990s. Today, Physeter macrocephalus is listed as vulnerable on the IUCN Red List, a category that reflects genuine population risk, not a bureaucratic formality. In the United States, the species is listed as endangered under the Endangered Species Act. The Mediterranean subpopulation carries an endangered designation from the IUCN specifically. Population trend, unknown. The animal whose muscles run near black with stored oxygen, whose chest collapses under 100 atmospheres of pressure, whose acoustic beam is the loudest sound ever produced by a living creature, whose social units have maintained distinct cultural dialects across thousands of kilometers for generations. That animal is classified as a species at risk. Not because of giant squid, not because of orcas, not because of any failure in its own extraordinary biology, because of harpoons, because of spermaceti candles and industrial lubricants, and the assumption that the ocean was inexhaustible. The sperm whale needed none of our attention to become what it is. It built a civilization in the dark, 1,000 meters down, long before we arrived.
The only predator that has ever seriously threatened it, the one that named it wrong.
If you want more stories like this one, subscribe.